Welcome people, I will be your commentator for today's video, and today I have a special one for you. Can you guess what it is? If you guessed meiosis then you are 100% wrong. Just kidding. Lol. We are learning about meiosis, so strap in your seatbelts and prepare to learn. Meiosis, a cell that is split up into two sex cells and then becomes four daughter cells that each have 23 chromosomes. These daughter cells are unique and are also haploid. This all happens during fetal development. Today, I'm going to be showing you the process of meiosis, which is all parts of the interface through the prophase. Anyways, the interface phase 1. There, that's better. Anyways, the interface phase 1, this is the resting phase between successive mitotic divisions of a cell. There are 3 stages of cellular interface, with each phase ending when a cellular checkpoint checks the accuracy of the stage's completion before proceeding to the next. The stages of interface are G1, gap 1, in which the cell grows and functions normally, synthesis, S, in which the cell duplicates its DNA, and G2, gap 2, in which the cell resumes its growth in preparation for division. The interface is basically the cell that spends the most of its life.
Wow. That's a lot.
Shut up Zero, no one cares about you or your knowledge. Oh. Get roasted. Ha 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 ha. Let's get back into this project. Next up, the prophase. It's known for when the chromosomes become visible as paired chromatids and the nuclear envelope disappears. The first prophase of meiosis includes the reduction division. It is also the first stage of cell division in both mitosis and meiosis. Beginning after interface, DNA has already been replicated when the cell enters prophase. The main occurrences in prophase are the condensation of the chromatin and the disappearance of the nucleolus.
May I do one?
You know what, Zira? That's fine, but make it good.
You can always count on me. Hey, what's up guys? My name is Zira and I'm going to be doing the metaphase. The metaphase is the second stage of cell division between prophase and anaphase, during which the chromosomes become attached to the spindle fibers. The chromatids line up along the equator of the cell. Once aligned, one fiber from the north pole of the cell and one fiber from the south pole of the cell attach to each sister of a chromosome pair.
Holy shit, Zira, that was amazing. Where did you learn this from?
I learned it from you, boss.
If you want, Zira, you can do one more, only because I like you. And by the way, I'm very sorry for roasting you, a liar.
Oh okay, apology accepted. I would gladly do another one just for you. Welcome back. For my last one I'm going to be presenting the anaphase, which is basically the metaphase continued. The anaphase's fibers pulls the homologous chromosomes toward opposite ends of the cell and result in creating a structure called a tetrad. Also during the anaphase the cells grow larger and prepare to divide for meiosis, to which we will come in contact later if you know what I mean. LOL.
Wow Zira, you are the best. Please come visit me anytime you want, because you are awesome. We are now moving on to the last part of meiosis 1, and that is the telophase and cytokinesis. These are two nuclear membranes that form and separate into two cells. During telophase, the chromosomes begin to uncoil and form chromatin. This prepares the genetic material for directing the metabolic activities of the new cells. The spindle also breaks down, and new nuclear membranes form, and then during cytokinesis, the cytoplasm splits into and the cell divides. Cytokinesis occurs somewhat differently in plant and animal cells. In animal cells, the plasma membrane of the parent cell pinches inward along the cell's equator until two daughter cells form. In plant cells, a cell plate forms along the equator of the parent cell. Then, a new plasma membrane and cell wall form along each side of the cell plate. Did you get that? Cause if not I will, but notes on the screen if you didn't. Tell me when you're done. I'll wait. Lol. I'm so silly I can't even hear you guys. My bad. Well anyways, moving on. The first step of meiosis second begins with the two daughter cells produced by the first meiotic division. As in prophase, the chromosomes are condensed. During this stage of spermatogenesis, the cells are called secondary spermatocytes, or during oogenesis, secondary oocytes. The chromosomes are not yet attached to the spindle apparatus, which is growing outward from the centrosomes. In metaphase 2, the chromosomes line up in a similar way to the metaphase stage of mitosis. The chromosomes become arranged on the metaphase plate, much as the chromosomes do in mitosis, and are attached to the now fully formed spindle. This is in contrast to metaphase 1, in which homologous pairs of chromosomes align on the metaphase plate. Anaphase 2, the sister chromatids separate and move toward opposite ends of the cell. The separated chromatids are now called chromosomes in their own right. As the chromosomes are dragged along by the spindle apparatus, their arms can be seen dragging along behind, so that the chromosomes form V-shapes. Telophase and cytokinesis 2, a nuclear envelope forms around each set of chromosomes. Cytokinesis takes place, producing 4 daughter cells, each with a haploid set of chromosomes. Because of crossing over, some chromosomes are seen to have recombined segments of the original parental chromosomes. Well, that's all folks, hope you enjoyed the video. This took forever to make, but it was all worth it, at least I hope so, and I apologize for my annoying voice. Hope I didn't give you internal damage, lol lol lol.